Scientists have made use of exquisite sensitivity and specificity of antibody recognitions for use in assays for both diagnostic and basic research applications. Technological advances and methods to generate antibodies and to detect antibody-antigen binding have transformed laboratory medicine and basic research in many fields. So in this chapter, we will be doing the introductions of diagnostic applications of immunology. The first test is the serological assays. So what is serological assay? It is a laboratory test that checks for the presence of antibodies or other substances in a blood sample. Reminded that positive and negative control are mandatory for a proper interpretation of result for a proper diagnosis of a disease. The next test is the classical assays. Classical assays have two. It is called immunofluometry. Immunofluometry is a preferred alternative for a gel-based precipitation assays. It is also based on the formation of antigen-antibody complexes and make use of antibody and or antigen calibrators. The analyte of interest is measured by light dispersion. That is why it is called nephelometry. Next is immunofixation. This test is used to identify proteins called immunoglobulins in our blood. So the next test is the Western blot, or it is called Western blotting. It is a technique that identifies a specific proteins in a given sample after their separation using polyacrylamide gel electrophoresis. The polyacrylamide gel is placed adjacent to a membrane, which is typically intracellulose or PVDB or polyvinylidene fluoride, and the application of electrical current induces the proteins to migrate from the gel and immobilize on the membrane. Let's move on to the agglutination assays. It is used for a variety of purposes in bacteriology and diagnostic immunology. Next is bacterial agglutination. It is observed when a bacterial suspension is mixed with antibody directed to the surface of determinants. The third is the agglutination of inert particles coated with antibody or antigen. Latex particles and other inert particles can be coated with a purified antigen and will agglutinate in the presence of specific antibodies. The last one is the red cell agglutination or the hemagglutination. Red cell agglutination is the basis of a wide array of serological tests that can be subclassified as direct or indirect hemagglutinations depending on whether the assays involves a single step or two steps. The next test for immunology is test based on the immunofluorescence. So it has three. First is direct immunofluorescence. It uses a single antibody that is chemically linked to a fluorophore. Next is the indirect immunofluorescence. It uses two antibodies. The unlabeled or the primary antibody specifically binds the target molecules, and the secondary antibody, which carries the fluorophore, binds to the primary antibody. The last test is the immunofluorescence test in microbiology. It can be used to identify isolated organisms, to identify infectious organisms in tissue biopsies or exudates, and also to diagnose an infection through the demonstration of the corresponding antibodies. Let's move on to the monoclonal antibody-based assays. So the production of the monoclonal antibodies involves two cell populations and three major steps. First step is fusion, second step is selection, and the last step is screening. Most commonly, spleen cells from a mouse immunized with antigen of interest are fused with transformed murine plasma cells. This fusion results in the formation of hybrid cells or hybridomas that will proliferate like transformed cells. The B cells hybridomas will produce the same antibody as a parental B cells. The next is the flow cytometry. Flow cytometry has become one of the most important and powerful technologies for clinical immunology, basic research, and drug development. A flow cytometer is an instrument that measures cells in a fluid system or a fluid stream. Within the flow cell, cells pass in a fluid stream in front of a laser light source, which allows for the measurement of multiple properties from individual cells. In cell isolation, some flow cytometers have ability to collect population of cells or individual cells expressing a given phenotype. The sorted cells are used for a given or variety of downstream applications that include functional analysis and molecular profiling. Cell isolation or cell sorting is very common in a research setting but is not used in clinical laboratories for patient care and treatment. Next diagnostic test for immunology is immunohistochemistry. Immunohistochemistry is a result of the application of monoclonal antibodies to the detections of antigens in tissues and has replaced immunofluorescence in many applications due to the specificity of MABS. What distinguishes the techniques from flow cytometry is that the cells are evaluated within their physiological context because the tissue remains in its original microenvironment. Okay, next is a highly sensitive immunoassays. The use of monoclonal antibodies and specialized readout systems have led to generations of highly sensitive immunoassays that replaced older platforms such as the radioimmunoassays. Lastly, the enzyme immunoassays. The enzyme linked immunoabsorbent assay or ELISA, currently known as the enzyme immunoassays, is a flexible platform for the detections of an unlimited number of antigen in a variety of matrices. So those are just a glimpse of the overview and then introductions of the chapter 15 of the diagnostic applications of immunology. I do believe that in medicine time, these things will be clarified to us and will be discussed thoroughly through the procedures. But this time, it was solely on the introductions of the test. Thank you so much.